Hi everyone, welcome to Faucet's Molecular Machines Group. Really delighted to have Sander Wezenberg here today. And Sander will be discussing artificial molecular machines operating in the lipid bilayer. Really excited about this presentation. I already shared a bit of info about you here in the chat, and I'll also share a link to your bio here in the chat for people who want to follow up. With that being said, the stage is yours. Really excited for the presentation, and I'll monitor the chat for the Q&A. Thanks for coming.
Yes, thank you, Alison, and sorry for taking a while. It's very nice to see friends and colleagues in this room, and thank you for joining. Today, I would like to discuss the operation of molecular machines in lipid bilayers, and I will show some work that we did on using molecular receptors that are responsive to external stimuli that can be used to control transport processes across membranes. But this is a foresight lecture. I thought it would be appropriate to start with this slide that, of course, everybody is familiar with. And it's a famous lecture of Feynman in which he discusses the possibility of moving atoms or molecular fragments in a way so that you can synthesize chemical substances. And of course, for that, you would need molecular machines. And it took some years before Eric Drexel started to further developed this idea and termed this molecular nanotechnology, or more specifically, molecular manufacturing. So what Feynman also mentioned in his presentation, and this was an idea given by his friend Albert Hibs, it's that it would be very interesting in surgery if you could actually swallow the surgeon. And maybe small machines could be permanently incorporated in the body to assist organs that are not functioning properly. And of course, this sounds very science fiction and may remind you of some cartoons that you have seen in your childhood. And often when I talk about the idea of using molecular machines in biology or applying them in biology, one of the main questions often is, so how are you going to make them water soluble? And my answer usually is I don't think it's necessary, because in biology, there's many hydrophobic compartments. For example, the lipid bilayer that makes up the cell membrane. And this lipid bilayer, it's filled with all kinds of proteins. And in particular, transport proteins that are able to mediate the passage of solids across this membrane that otherwise would not be able to permeate. The assistance of proteins in this transport is really, really important for many biological functions. For example, to maintain osmotic pressure, for metabolism, and for signaling. So the question is, of course, because of this important role, defects, in particular genetic defects in these membrane transport proteins, they are related to very serious diseases. The question is if whether we would be able to make fully artificial systems that can achieve similar functions as these proteins. And then at first we might be able to use them to interrogate biological systems, but maybe further ahead in the future, hopefully we could make fully autonomous systems that are able to take over the function of such malfunctioning proteins and thereby act as new kind of pharmaceuticals. One of the most widely known disease related to faulty transport is cystic fibrosis. Originally, the symptoms of this disease were already widely known. They lead to salty skin and damage of pancreas. And in the Middle Age, this was ascribed to children being bewitched. And it was actually in Leiden where the first autopsy on a child was performed by Peter Pauw, where he described for the first time in detail the symptoms of this disease. And this was done in the anatomical theater in Leiden, and you can still see a very nice reconstruction of this anatomical theater in the Boerhaave Museum in Leiden. So that's a very nice suggestion to go when you would be able to visit this. So now we know that cystic fibrosis is related to a faulty chloride channel, this CFTR conductance regulator, which you see on the right. And this is a channel for chloride. And what basically happens is that because of different types of genetic mutations, the opening and closing of this channel in response to phosphorylation, it's not working anymore. So in our group, we're interested in developing molecular receptors that you can switch between different states and thereby control binding affinity to these molecular receptors. And on the one hand, we hope we can apply such receptors to control transport processes in membranes. On the other hand, we try to use them to improve extraction processes, for example, of toxic anions that are present in the environment. So the latter I will not talk about today. But so a very simple idea, which was already developed in the 80s by Shinkai, it's based on these type of molecular tweezers, where you have a receptor with two different binding motifs, and upon application of a stimulus, in this case light, you bring those binding motifs together, and thereby you elevate the binding affinity. In principle, we're interested in using a different type of stimuli, but we started using light because it's relatively easy to apply and it doesn't produce any waste in your system. So the examples I will discuss today are all light-based. We became inspired by this work of the group of Phil Gill and Tony Davis, among others. They developed non-switchable molecules based on ureas or on conic spiral motifs, and they showed that these are able to mediate transport in the lipid bilayer membrane. So you can deliver them to the membrane or pre-incorporate them. They like to be resided in the membrane because they are in the hydrophobic environment, but they are mobile, and thereby they can pick up anionic substrate on one end of the membrane and mediate its transport over the membrane. But that's, of course, all down a concentration gradient. So it's driven by concentration gradients. But this feature gives them some very interesting biological properties. But of course, when you look at proteins in nature, these transport systems are all regulated by external or by stimuli. And a very nice example, it's this nitrate transport protein, which you find in plants. And when the nitrate level in the soil is very low, it's phosphorylated, gets to a high affinity state, while when the nitrate level in the soil is very high, it's dephosphorylated and goes to a low affinity state. And actually, in that way, the cell can control the concentration of nitrate within its interior. But it's not only chemical stimuli that you see in nature. So there's a number of proteins which you find in bacteria, which mediate transport and are controlled by light. Examples are these anion and cation channels, which have retinol embedded. So retinol can be switched by light and thereby induces conformational changes in these proteins. And this can open and close channels. And maybe more interestingly, it's that they can also be used to create concentration gradients. I think that's very interesting because in that way, you're converting light energy into a concentration gradient across a membrane, which you could regard as an energy reservoir. For example, in bacteria rhodopsin, then the protein gradient that it's created can be used for ATP synthesis by ATP synthase. Also very interesting to mention is that nowadays these type of proteins are frequently used in the field of optogenetics. In optogenetics, you can induce and control the activation of neurons using light, and they use these type of proteins for that. But the drawback is that you need to express those proteins by mutations and also often have to feed retinol to for example, mice that you would like to do these studies in. So in our group, we're interested in developing fully artificial systems based on molecular switches and molecular machines with which you would be able to achieve similar kind of functions. Also, other groups are active in this field. For example, the group of Amar Vlad, he developed these very nice foldamers based on azobenzenes, with which are able to control binding affinity. And also the groups of Yeong and Langton, among others, they developed these tweezer-type systems with urea functionalities. And by switching with light, you can control binding as well as transport activity. In our group, we focus on the use of stiff still beam switches as the core. And that's because stiff still beam, as you see here, it has a very rigid core structure. It undergoes a very large change in geometry when you switch it. And we think that's very important for creating large differences in binding affinity. And also, in contrast to azobenzene, which normally thermally relaxes from the photogenerated Z isomer to E isomer, these switches have two thermally very stable states. So that allows us to synthesize them, isolate them independently, and to study their properties separately. So one example, we introduced these urea groups to these switches, and by NMR and UV-Vis spectroscopy, you can very nicely follow these isomerization processes. So of course, when you start with the E isomer, you radiate with UV light, you see typical signals appearing in the NMR spectrum that belong to the Z isomer. And in our case, we generate around 50% of the Z isomer at the photostationary state. And then by irradiating at a bit longer wavelengths, you can again go back to the E isomer, and you can do this over and over again. We studied binding properties by NMR titrations, and already when doing job plot analyses, you see very distinct binding modes. So you see that for the E isomer, you get 2-to-1 binding. So one anion can bind to either side or to either urea on the molecule. While on the Z isomer, the anion can bind between the two binding motifs. And then this leads to much stronger binding to the Z isomer than to the E isomer. We tested that for a range of anions. So apart from chloride, you see particularly strong binding of acetate and dihydrogen phosphate. In all cases, much stronger for the Z isomer than for the E isomer. You can also calculate these structures. So DFT supports this 1-to-1 binding in the Z isomer. Here you see that for acetate and dihydrogen phosphate. For chloride, we were not always completely sure because it binds a lot weaker, and job plot analysis was not conclusive. But however, we did observe this 1-to-1 complexation in the solid state by X-ray analysis. So here you very nicely see these 4 hydrogen bonds from the urea NH protons to the chloride anion, and one of them is slightly longer than the other 3, and that it's because in the crystal packing it also forms an intermolecular hydrogen bond to a neighboring molecule. But then the main question was, could these type of molecules, could they function as carriers in membranes and thereby mediate transport? And we already started studying that some years ago in collaboration with the group of Phil Gill, who has a lot of experience studying membrane transport of anions. And currently also all these transport studies, we do them in our own lab in Leiden. But most of the work I will present today was done in the lab of Phil at the University of Sydney. So we made different derivatives. Overall, they have very similar properties as the first molecule I showed you. So PSS ratios by light irradiations and so are very similar. Binding properties are very similar, except for the nitro-substituted compounds, which have very poor band separation and therefore cannot be switched back and forth. So a way to screen the transport activity, it's by doing these HPTS assays. And what you see, just the gray circle, just a lipid bilayer composed of PUPC lipids. and you can encapsulate a HPTS pH-sensitive dye and load it with sodium chloride and buffer it to pH 7 and have the same sodium chloride and buffered solution on the outside. Now, after adding your receptor, you can apply a base pulse, so you get a pH gradient over the membrane. And there's two ways that this pH gradient can be dissipated. One is by HCl symport, and the other by this chloride hydroxide antiport. And in both cases, then the dissipation of this pH gradient, it's a measure for your chloride transport. And when you do this, you already see very distinct differences in activity between the E and Z isomers, as you can see in the graph below. So you can express this in half maximal effective concentrations. And when you do that, you see very, very large differences in activity between the E and Z isomers, actually much larger than what you would expect only based on differences in binding affinity. Now this HCl symport and chloride hydroxide antiport, it's not a full story. So sometimes in some transporters, they are very selective uniporters. So they are only able to take a chloride ion over the membrane. And there's a very simple way to test that it's to add a protonophore, so something that mediates the transport of the proton. And in case the protonophore would lead to enhanced transport, that means that your main mode of your main mechanism is a uniport process. And in our case, we found this for the non-substituted urea and thiourea compounds. So we see a big increase in activity once a protonophore is added, meaning that it follows a uniport process. Now, as you have a receptor that you can switch between states with distinct transport activity, of course, you can do exactly the same in situ. And here we did a similar assay, but it's based on scattering. So this is called an osmotic assay, in which the size of the vesicles changes slightly upon transport of chloride out of the vesicles. And then this you can measure by a change in light scattering. And we do this because sometimes the HPTS fluorescent dye interferes with irradiation of our molecules. And what you can very nicely see here, also the y-axis is a measure for the transport. And you can start with the E isomer, so not much happens. And then you activate transport by isomerizing to the Z isomer. You can partially deactivate it again and activate it then by irradiation. And of course, it's not completely perfect because the photoconversion, it's not fully quantitative. And also still the E isomer has some activity. What it's also very nice is that because our transporter follows a uniport process, we're also able to depolarize membranes. So what we did in this case was to incorporate something in O dye, which is a fluorescent dye with which you can follow membrane potential. And now we loaded the vesicles with potassium chloride. We have a sodium chloride external solution. And we first add a potassium channel for linomycin. So, and because of the potassium efflux, now a membrane potential is built up. And then after adding our receptors and activating them by light, you induce a chloride uniport, and with that, the buildup of a chloride gradient over the membrane. Now, of course, the gradient here, it's not driven by light, it's just the dissipation of the gradient, it's activated by light. The gradient itself, it's built up because we start with a cation gradient in our vesicles. But overall, we showed very nicely that apart from controlling transport activity, we can also control membrane depolarization. So to some extent, there's some similarity to the anion channel rhodopsin compounds I showed you in the beginning of the lecture. Not yet halorhodopsin because, of course, the creation of the gradient, it's not driven by light itself. We've also made many different derivatives and tried to see if we could optimize these transport processes, but many of our designs turned out to be not so active in transport, and it still turns out to be very difficult to design new transporters. And I think that's also one of the main reasons why many examples you find nowadays in the literature, they are based on existing designs and not so many fully new designs. One example I wanted to highlight is this stilbene receptor based on bisamidopyrrole. Also in this case, we expected that actually by bringing the binding motifs together, we would have enhanced binding. But now we do have a little bit more overcrowded system where we have these methyl substituents to stilbene, and somehow that leads to a slightly larger dihedral angle for the Z isomer. And we found in this case that actually in both cases, for both isomers, we do get 2-to-1 binding without any significant difference in binding affinity. Nevertheless, still, by testing them in transport, there's still significant difference in transport activity. So again, you can still use them to activate membrane transport using light. But it's very interesting. It very nicely highlights that binding affinity, it's not the only parameter we have to think about. It's also about solubility in the membrane and mobility in the membrane. But still, when we started, we always thought that one of the main things we had to do to control membrane transport was to control binding affinity. And therefore, we looked at these calyx spiral systems, which were developed by the groups of Lee and Sessler, and they introduced these bridges and thereby found that these bridges create a very nice binding pocket so that you get high affinity for chloride binding, where all these pyromoieties can point upwards and form hydrogen bonds with the anion. So we decided to incorporate our stilbene molecular switch into these bridges, and thereby expected that in the Z isomer we would have a significant chloride binding, and chloride binding would be weaker in the E isomer. And of course, you can monitor these processes by NMR and UV spectroscopy. And in this case, you can switch quantitatively from the Z to the E isomer, and then partially back again to the Z isomer. And all of this shows similar UV vis absorption profiles as before. In binding, it behaves slightly different. So once you start adding chloride, you see initially both bound and unbound species in the NMR spectrum. So that means that the exchange between bound and unbound species, it's slow on the NMR timescale, but then upon adding one equivalent, it's already fully saturated. So all the chloride is bound. Therefore, we determined binding constant by ITC titrations. And you see that the binding, it's quite large and very similar to the binding affinity that was previously reported for the systems that were developed and reported by the group of Sessler. When we did similar studies for the E isomer, I was a little bit surprised initially. So when we started adding chloride to the E isomer, we did not see any changes in the NMR spectrum when we performed the studies in DMSO. So we did expect an affinity difference, but we still thought some binding would happen to the E isomer. But it does not. It did not bind at all to the E isomer. And also when you do ITC studies, you see only very minor heat release, not significant. And eventually we were able to determine a binding constant by saturating it in a NMR titration in acetonitrile. And by fitting all these data, we found a binding constant which is around 8,000 times lower than what we found for binding to the Z isomer. And this is really, really a large difference. So it's about 2 orders of magnitude larger than the best differences that we were able to achieve and others were able to achieve in photoswitchable anion receptors. By X-ray analysis, we got some insight into the binding motif. So for the Z isomer, you very nicely see that the molecule adopts this cone conformation, where all the chiral unit points upwards and hydrogen bond to the chloride ion. And besides, there may be some CH anion additional interactions. For the E isomer, you see there's very few space to bind the chloride. Furthermore, we also considered inverted type cone structures, which would still maybe bind chloride. But in general, the calyx spiral prefers to have an alternate conformation in which half of the pyrals point upward and half downward to minimize the dipole moment. And then upon binding, the binding energy is sufficient to compensate for this the energy that this conformational change cost. But we found that in this E isomer, there's actually a lot of strain on this bridge. And by DFT calculation, it seems that therefore it costs a lot of energy to adopt this cone conformation. So we think that's one of the main reasons for this low binding affinity. Also, usually the E isomer of stiff stilbene would be flat, and here you see that it's somewhat bended, illustrating that there's strain on this bridge. So lastly, I would like to comment a little bit on the irradiation wavelength. So in the examples I showed, we use high energy UV light, and for biological applications, it would be beneficial to move more to the visible light region or maybe even the near IR region. And there's different strategies that you can use for that. We've also worked on these strategies. I want to show one example today where we introduced push-pull substituents. So in this case, we introduced this dimethylamino and cyano substituent. And because of that, the absorption wavelength, it's slightly redshifted. And with that, you can just use a longer wavelength light to isomerize them. I think another interesting part that we found here is that the fatigue resistance, it's also improved a lot by using visible light. Now, of course, when you protonate this dimethylamino substituent, you break this push-pull system or this push-pull interaction, and thereby you again get a very big blue shift. And of course, you cannot isomerize the molecule anymore using visible light. But what we also saw is that now when you would go to the Z isomer and in the presence of acid, now the Z isomer thermally relaxes to the E isomer. And that's a little bit surprising because as I mentioned in the beginning of the talk, normally these stiff stilbene switches have very, very high energy barriers for thermal isomerization. But in this case, for some reason, by acid, we can catalyze this process. And this was known for stilbene, but was usually performed in concentrated sulfuric acid. And in our case, we can just use a little bit of TFA, so a mild acid, to induce this process. And it gives us some additional control over the switching. And at the moment, we're also developing other systems where by using protonation, we can control both thermal and photochemical isomerization processes. So lastly, I would give some of the challenges that we face and things we would like to look at in the future. And I think the most important point is to go from facilitating diffusion, so transport from high to low concentration gradients, to go to active transport. So to be able to build up gradients by using, for example, light energy. In all the examples reported so far, where molecular machines or receptors are embedded into membranes to control transport, this is always passive transport. Another aspect, it's most studies still are performed in model systems, so just in model liposomes. It would be nice to go to cells to try if we can mediate this transport also across cell membranes. A very difficult issue there, it's still that in general, we like our compounds to be hydrophobic and lipophilic so that they enter the membrane, they can be delivered to the hydrophobic membrane. But on the other hand, that means that once you add them to an aqueous solution, they may quite rapidly precipitate. So there's still some work to be done on deliverability and targeting strategies for that. At the same time, that also exposes a little bit a problem in the studies that we have, and that it's that often we, in transport assays that are done, we look at anion transport, and based on the rate of this transport, decide whether we have a good transporter or not. But when we do not see transport, it may also be the case that the molecule is poorly delivered or poorly incorporated. And we've now found several examples of that in our group, where actually by pre-incorporating them, so by preparing the liposomes when the molecule is present already in the lipid film, actually you do see activity. And I think that in some cases may be a little bit misleading us in developing switchable receptors, that we think that there's an activity difference, but actually we are looking at a deliverability or incorporation difference. Further, we would like to move from light to other stimuli, for example, using chemical stimuli to control these processes, or using both of them orthogonally. Another thing we are working on, it's sometimes we consider our molecular receptors that are responsive to light as a kind of signal transducers, because basically what happens, it's we have a receptor that binds a substrate. When we shine light on it, the substrate is released, and we are trying to use this release and uptake of these substrates to couple this to other processes, for example, to falling processes or catalytic processes, so we can remotely control these processes. And one of my PhD students, Indigo Beekaerts, he will give a VMASC lecture about this on the 30th of March. Lastly, what I would like to mention is that 2 years ago, I developed a course on molecular nanotechnology at the University of Leiden for master students. And if you would be interested in developing a similar course at your university, then please write me, and I would be happy to share some lecture material or to discuss the course and improve the course further. I would like to thank my group, in particular David and Jasper, who were involved in the projects I presented today, as well as the collaborators mentioned in the right bottom. I would also like to thank the funding agencies, and I would like to thank you for your attention. Thank you.
Thank you.
Wonderful. Thank you so much. Really, really appreciate it. Thank you for all of the wonderful insights. You're getting lots of virtual claps from the chat. Thank you. Do you mind if we already dive into questions, Sander? Because we already have a few in the chat, and I'm assuming that we'll have a few more collected by the time we get through them.
No, I'm happy to start. I will keep my slides shared so that maybe I can go back to a slide if it's necessary.
It sounds good. Reto, do you want to unmute first and go shoot away?
Sure. So one thing I've been looking at in this area is carbon capture with light-sensitive release, because I figured that's something we could actually build. And so I was wondering if you looked at that as well.
No, so no, we have not looked at that ourselves, but that would be a very interesting idea. We have more thought about, for example, capture of toxic anions, or also Kallik spirals known to strongly bind ion pairs of cesium halides, for example, So currently we're working on a extraction of radioactive cesium salts, and thereby could again deliver them to smaller volumes to reduce waste streams. But no, carbon capture, not yet, but it would be also nice.
Do you think the shape is like the charge difference and stuff on the carbon is significant enough that you can actually grab onto it with one of your pliers?
Yeah, the selectivity. So
Right.
that it's always a difficulty. You can, of course, in the tweezer type approach, you can further functionalize them. It also gets a little bit more synthetically more complicated. So in the case of macrocycles, actually, like the Calyx spiral, it's a little bit easier to achieve high selectivity. Yeah.
I have another one. Like, what's the fastest switching speed you have actually observed where you can reversibly switch on and off? Like that you can actually bind something and, like unbind it. Like I figure that should be in milliseconds, but I don't know if you can actually measure it with NMR. And is it one that has like an on frequency and an off frequency, or is it one that's like monostable?
Yeah, so that's a very good question. So the photo switching, it's really, really fast. And also in our case is the substrate binding, it's also dynamic. So in the picture, you present it like you bind and release something, but what you're actually doing, it's changing affinity, and it's in dynamic exchange. So we do not have such a good insight in how these relate. We do have examples where the binding has influence on the photochemical switching, but we need to do much more studies for that to get more insight. I think the latter system, where the binding itself, it's slow on the NMR time scale, it gives us the option to get much more insight into binding kinetics and also how that influences switching as well as transport. Yeah, I hope that answers your question.
That's really good. Thank you. What's the difference in KD that you get when you switch?
I always think in KAs, so it's the inverse. But for the tweezer type, it's around 20 to 50 fold. And for the macrocyclic structures we get differences in Ka around 8,000 fold.
All right, Richard, you happy for now? Then we're moving on.
Thanks.
Great. We're moving on to Tomo next.
Thanks. Yeah, so I came from a optogenetics and neuromodulation background, and one of the big problems has been using light to change things in a larger animal just due to the delivery volume being limited. And a lot of research is now being moved towards kind of modifying, say, ligand-based receptors, temperature sensitive, ultrasound sensitive, or magnetic sensitive. And how easy is it to just switch the switching mechanism with the basically the pore design or the kind of the transport area design? Like, are they almost at a level where it's like a toolbox? You could say, I want to do this with ultrasound energy. I'll use this backbone and then this transport.
Yeah, that's a very good question. In case of ultrasound, my expectation is that it's not so straightforward. I think ultrasound, it's often just some local heating that maybe you improve or you generate an effect.
So there are mechanosensitive ion channels like piezo, but there are also heat sensitive like TRIP, and you can actually use both to non-invasively change effects deep in tissue. So it's one of the things. So I think ligand based probably gives you maybe the most orthogonality and least amount of external equipment for it to be clinically translatable. Like you can move from a rat to, say, maybe a pig model, and you just have to give it something in the feed without anything attached to them.
Yeah. Yeah, so originally we started by making light responsive receptors, as I said, because it's relatively so many molecular switches are known, so they are based on molecular switches. We can apply it relatively easy. We do have interest in making more allosteric kind of systems that might be responsive to internal stimuli. We have not thought about other types of stimuli yet. Yeah.
Yeah. And there's probably a bias in where I'm approaching from, but with a delivery, are you just using passive methods or are you applying energy like thermal or mechanical? Because that tends to help. But then if you want to stabilize them like in a membrane as opposed to precipitate out, having like anchoring to cholesterol or other portion, it's going to be very chimeric at that point. But I'm just thinking like in 5 years kind of thing. Those types of things can be used to prevent precipitation or misforming, like kind of stabilize their shape so that they can remain effective for a longer time.
Yeah, so that's a very good point. So indeed, with some non-switchable systems, people have, for example, used water-soluble cavitants that could encapsulate your receptors in the way to deliver them. We've also thought ourselves more about like dextran or micellar structures could be used. I think that's a very good point, and it's something that still needs a lot of investigation. Anchoring to cholesterol, we've considered that, but we have not worked on that yet. Yeah.
Okay.
Great. Love it. A new research direction then. Anything else to know, or you good?
That's it. Thank you.
Awesome. Wonderful. Thanks for the suggestion. Next one up, we have Stefan. Stefan Bosny, if you want to mute.
Hi, Sander. Thanks for the talk. Yeah, I was just wondering about whether you looked into whether the ion binding affects your photostationary state of your switches. And then I guess related to that, whether you'd considered what is rate limiting in your transport. So is it ion binding, diffusion across the membrane, or release of the ion? And to what extent does that change between your isomers, or is that just not something you've been able to look at yet?
Yeah, so that's a very good question. Thanks, Stephen. It's not something we've been able to look at yet. So we have looked at, for example, whether, because now we have a receptor which we just switch between an active and a non-active state. We've looked at the influence of continuous irradiation, how that affects transport. I think it's difficult to, whether it's the kinetics of binding or whether it's the mobility in the membrane. It's probably has to do with all the things you mentioned. It's hard to dissect it, in particular because for the tweezer type systems, it's not so easy to determine the kinetics of binding. It's all a very fast exchange. Yeah, I don't know. What are your thoughts on this?
I don't know. I'm just wondering whether there might be some interesting effects where one isomer has one process as rate limiting and the other one has a different process as rate limiting, whether that could be an avenue to explore. But I don't know.
Yeah, it is, of course. You can do various assays and you can investigate or you can get more insight into the mechanism. And it would, of course, be very interesting to see that also upon switching, the other isomer would not follow the same preferred mechanism. We do have some more recent insight where we do see that mobility plays a very important role. But I think it also, this is again very dependent on the system you're studying. Yeah.
Okay, lovely. Thanks a lot.
Well, we have about 6 minutes left. And so I'm going to the usual algorithm where I ask more general questions that are very foresight relevant. And you could stop me by asking another one. So usually one thing that I would always love to know from folks presenting is, I know that speculation is not something that you guys tend to do very easily in these scenarios, but I would love to know if you could maybe project outward in the future in a really good scenario where things go well and where progress is trotting along or perhaps even accelerating. Where do you think these capabilities could be in the next 5, maybe even 10 years? Like, what do you think are a few either exciting applications down the line or a few new capabilities that could maybe unlock a few other scientific areas? Like, what could we get excited for?
Yeah, I think what would be exciting, it's one of the things I mentioned, it's to be able to deliver them to cell membranes and in that way be able to interfere with biological processes by turning transport on and off. A very simple idea would, for example, to activate transport and disrupt ion homeostasis and thereby induce apoptosis, for example, and thereby you would have a light-based method to do that. Furthermore, I think in the moment you would be able to convert the light energy into generating gradients across membranes that would open up a lot of opportunities, because these gradients you can again couple to other processes and drive other processes. I think in general, so I think if you look more at applications in biology, so probably many of the drugs we have, they are always protein inhibitors. And I think actually by using systems in membranes and controlled transport across membranes, and in that way, someone mentions the work by Gast and also later by Bakker. So there's indeed a very nice work by Gast able to generate proton and calcium gradients across membranes by using a slightly different concept. So I think that would be very interesting, and also it opened a new way to interfere with biological processes, to do things in the membrane. Yeah.
Cool, thank you. And is there anything for someone who's perhaps more junior, either in the seminar right now or listening to this afterwards on YouTube, is there anything that you would recommend them investigating further? Any new research directions that you think are really exciting? Any challenges that you see blocking this work where you want to direct a bit more attention to?
I think in general, it's the often in the field of switches, often people address two states with different properties. But actually, when you irradiate a molecular photoswitch, you get a photostationary state, which is actually a photodynamic equilibrium. So you have a continuous interconversion between isomers. And I think it should be, we should harness that continuous interconversion for functions.
Lovely. And then the final question before we let you go is, if people now get really excited about your work in particular, what can they do to help you in your work along? Is there anything you're hiring, or you have a new collaboration that you'd like to pursue? Is there anything that folks can do to support your work in particular?
So I in particular, so to have more contact with the field of optogenetics. Fortunately, there's someone in the audience also. I think there we could benefit a lot from collaboration, and overall to work with people that are working on delivery strategies to membranes. There we could benefit a lot too.
Awesome. I really appreciate it. Thank you so much for coming online and for sharing your work with us. That was really exciting. Rachel just posted another index of our binding optical things. So as a tangential bit, at least. Thank you. We really, really, really appreciate it. This was fantastic. Thanks everyone for your awesome questions. I'm happy to be a liaison when following up with each other. So feel free to contact me if you can't reach each other. And thank you for presenting your work. It was a real pleasure.